I will start by presenting some slides. This talk is mainly about this bilateral collaboration between CIIMAR and the NIVA Institute from Norway. But also I'll be presenting some work that we've been doing on another project that is linked to this bilateral initiative, which is the SWAV project. And by the end of the presentation, I would like to show you a short 10 minutes video about fieldwork procedures for mapping the intertidal. So I just, as I said, this work started because we were interested in mapping seaweeds here at CIIMAR. And as you know, seaweeds assemblage include a variety of certain species that provide food, habitat, shelter for organisms from different trophic levels, therefore supporting complex trophic webs. Seaweeds are also considered a natural resource, not only for human consumption, but also as a provider of many compounds that are used in pharmaceutical and biotechnology industries. Therefore, there has been an increased demand for biomass by the development of these new markets. Because we were interested at CIIMAR in assessing the biomass, the standing stock of seaweed, we initially developed a project, which is the SWAV project, that intends to map the intertidal zone and assess seaweed biomass. This project came as an idea because we were doing previously some in situ work to assess biomass of seaweeds, and we faced several challenges. Some of them had to do with the time of the tides here in Portugal and the time that we had available to do the work, and to the extensive areas that we wanted to assess and map. So when we were also writing some other projects, we came across with another project, which is the CIBIO project from the NIVA Institute, that were also doing this sort of work, doing some mapping of the intertidal, not only for seaweeds, but also for counting birds and mapping seafloor, also counting mammals. But we had in common the use of drones or UAVs equipped with multispectral cameras. So that's why we came to the idea to merge these two projects, okay, and collaborate more intensively. And this was done in the scope of the call for the Fund for Bilateral Relations, from the EA grants, where we developed this UAV4SEA initiative, okay? This UAV4SEA initiative intends to reinforce bilateral relations between Portuguese and Norwegian scientists by the exchange of experience and knowledge in UAV remote sensing techniques applied to mapping coastal biological communities, not only seaweed, okay? And also it intends to promote future partnerships for joint research projects that we've been doing so far. A part of this bilateral initiative, we also intend to disseminate the acquired knowledge to the scientific community and stakeholders. So that's what I would like to do today, is to disseminate some of this knowledge to all the people that are involved in science today, and hopefully we'll collaborate for the management of coastal resources. Before jumping to the video, I would like to present now the SWAV project. This project, as I said, is the base of the UAV4SEA bilateral initiative on CIIMAR part. It started in July 2018 and has just been extended to December to 2021, this year, okay? It aims to develop methods for mapping seaweed beds and assess their standing stock based on two methodologies: RGB, multispectral, and hyperspectral images retrieved by drones, combined with spectroradiometer data collected in the field, okay? So just to say that this project has a team, of course, and I'm part of the team, but the PI of this project is presently Professor José Alberto Gonçalves, which I don't think is present today, but just to give you some remarks. How is the project organized? We have 6 tasks. I will briefly describe this diagram. So the first task was the exploration of the survey techniques for sites and the methodology selections, which has been done. We also, on task 2, collect the spectral signatures of the objects we want to classify, which were basically the seaweeds and different elements that I will describe later on this talk. Currently, we are doing task 3 because we had a bit of a delay due to the pandemics. That's why the project has been extended. And we are performing UAV flights equipped with RGB multispectral and hyperspectral sensors in 2 sites, in Vilachã and Viana do Castelo. In parallel, we are developing a plugin for the QGIS to assess remotely the biomass of the seaweed. In this case, the biomass is specifically for seaweed because that was one of our main interests. And in the meantime, we will have the validation afterwards of these techniques by ground truthing in the field. Transversal to this task is also task 6, which is the dissemination of the knowledge and skills transfer. Regarding the use of the spectroradiometer data, we wanted to see if the discrimination of seaweeds by their pigmentation was possible based on their spectral signatures. We know that seaweeds, either being red or brown, have different pigments, but we would like to see if the spectral signature would reveal that differences. So we collected several spectral signatures of seaweeds of these 3 dominant groups, the red, green, and brown seaweeds, with the spectroradiometer. And this was done in a black background to collect pure signatures of the samples, okay? What we did next, in collaboration with UTAD, University of Trás-os-Montes, was to perform linear discriminant analysis to see if there will be a segregation of these 3 groups according to their pigments. And in fact, that showed that seaweeds were highly differentiable in regards to the pigmentation typology. So you can see here that the green seaweeds were quite separate from the red and from the brown, according to their spectral signature. And also, there was some differentiation in terms of the reproductive status, although I don't have here the image to show you, okay? So this, we did it early in the project. And also, we wanted to know if the seaweed biomass variation would be different. If we wanted to know if different seaweed biomass would have different reflectance spectra, in order to be able to correlate later on the biomass with the reflectance spectra. So we did similar work as before, but in this time, we added and removed the blades to a 53 centimeter surface matching the ground field of view of the spectroradiometer. So when you have the spectroradiometer, if you have it to a certain height, according to the angle of the field of view, you will get a specific ground field of view. So we knew that placing the spectroradiometer in a tripod at a certain height, which I don't remember now what it was, but it was always the same, the ground field of view would be of 53 square centimeters. So the measurements ranged from one single blade to overfilling the ground field of view with seaweed blades by progressively adding several layers. Later, the seaweed samples were dried at 60 degrees centigrade for 48 hours to get their dry weight. The idea was then to build a relationship between the biomass and the NDVI. NDVI is a vegetation index that relates the near infrared and the red bands of the multispectral camera. I'm not insisting on this part of the talk on the differences between these bands. because there will be an explanation on the video about different bands that a multispectral camera has. In this case, the Mica Sense camera that we used for this work. So as you can see, the relationship is logarithmic, and it gets to a point when the biomass is, when it's too big, it gets saturated, okay? So this is probably an issue if you get big biomasses, because the readings on the NDVI are very close to 1, and even if biomass gets higher. In parallel to this work with the spectral radiometer, we've been performing since 2018 some flights, some UAV flights equipped with RGB and multispectral cameras. So today I'm going to present some preliminary work from the beginning, okay? And these data were acquired in February and May 2019 at Bilateral Rocky Shore. These flights were performed at 20 meters height and giving pixels of 0.8 for the RGB and 1.6 for the multispectral images collected. So all the images were precisely georeferenced using a high accuracy GPS receiver, were also auto-ratified, mosaiced, so that the data from different sensors, like the two ones that we used, RGB and the multispectral, and also data from different seasons, can be overlaid and compared in a geographic information system. The NDVI was calculated from the multispectral data and was used for mapping the intertidal and also used as a proxy for the seaweed biomass. So I have here now, okay, sorry. I thought there was the slides before, after, but it's still. So how did we do the mapping? For the mapping, we use supervised classification techniques applied in QGIS to evaluate the extension of 4 ecosystem elements, or which were seaweed, sand, mussels and rock, barnacles and limpets. So these were the categories that we wanted to map and assess in terms of area. To do that, to apply the supervised classification techniques, we need training areas. So these training areas we used were quadrant frames of 50 by 50 centimeters. And in this initial work, we used 3 quadrant frames for each of these elements, and an extra 4th one for the validation process. We now don't do this like this. We have increased the number of quadrant frames per class of object to classify, because we found out that the more samples you have for the training areas, the higher will be the accuracy of the classification. So we now use 70% of the quadrants for training and 30% of the quadrants for validation, which has increased quite significantly our accuracy of the classifications and of the maps, okay? So here is one of our colleagues getting the coordinates of training area. In this case is mussel and rock. And here on the right are the different objects that we wanted to classify, which were mussels and rock, seaweed, sand, and rock barnacle limpets at the bottom, okay? So this is overview of the shore from the RGB data. Sorry? Sorry.
Por favor, desliga o microfone.
Okay. Okay, carry on. This is a view of the Bilateral Rocky Shore during low tide, before we did the work, okay? At this point, probably there were already the training areas, because the quadrants, but you cannot see them here. I don't think you can see them from here. And this is the area where the multispectral camera flew, okay? So this is our region of interest. We have the data for the multispectral, and in this case, this is the NDVI, okay? And the NDVI can be quite high, here represented in white spots, okay? So this is a rock pool here, and that's why it says high NDVI, because it has seaweed. and the sand and water will be more dark, being water darker than the sand, okay? This is another overview with the location of the training areas and validation areas. Just a full extent of the training area. So as you see, you have rock barnacles and limpets, alga mix, which is the seaweed, sand, and we would have here 4, which is 3 for classification and 4 for each class. 3 for classification and 1 for validation. So this gave us some maps, which are here. The one on the left is from February, and the one on the right is from May. And as you can see, the 4 classes are here in the legend. The seaweed, or alga mix as we called it at the beginning, is in green. And from February to May, you see that rock pool here that I've shown you before. There were few seaweeds in February, but in May, the seaweeds have increased in covered area. Also, a little bit of the mussel and rock has increased. And this gives us some insights about the seasonal effect on the seaweeds. In fact, this supervised classification of the multispectral images from different seasons reflect the expected seasonal growth of seaweed beds and gave insights on the variation of the relative percentage of the main benthic invertebrates. So this information is currently being applied to the development of a QGIS plugin for remotely assess and map seaweed biomass. And hopefully this will be a valuable tool for rapid mapping and seaweed beds and quantification of their biomass, improving the knowledge of local ecosystems and contributing for the efficient and sustainable management of these coastal habitats. So this was it for now. And you have here the link for the video, because after this slides presentation, I would like you to please see a video of 10 minutes that we have assembled regarding the field work with drones for mapping these coastal environments. Okay.